Welcome to Unit 4, Lecture 3. So in this lecture, we're going to focus on the organelles that are involved in manufacturing and breakdown, and then also involved in energy conversion, which we already kind of touched on with photosynthesis and respiration. The control of the cell comes from the nucleus, and that's the big control center. You guys probably talked about that in middle school. So that's the brain of the cell. This is where we actually have the DNA, and then the DNA actually runs everything else in the cell. And the DNA gives information or gives directions to a lot of the rest of the cell to do different stuff. So the big thing is we end up having the nucleic acids. Most commonly we think of that as DNA, and then we actually do that to help make other stuff. All right, so the DNA actually directs everything else that's going on. So inside the nucleus is this little thing called the nucleolus, which is specifically created to make ribosomal RNA and ribosomes. Now ribosomes are really important because they actually make proteins. And we're going to talk a little bit more about proteins and how they basically do everything. Your entire body is basically run by proteins. So the DNA tells the proteins what to do. The proteins basically do it. Okay, and so there are two types of proteins. There are free proteins, which are basically hanging around in the cytoplasm. And then there are bound proteins. So these bound proteins are actually attached to the surface of something. And ribosomes can go back and forth. It's not like, hey, you're a free protein or you're a free ribosome. You're always going to be a free ribosome. Sometimes they're free ribosomes and then they become bound. Sometimes they're bound and they become free. So it depends on what it's doing, what kind of protein it's making at the time. So the endomembrane system includes a lot of pieces inside a eukaryotic cell to basically make stuff that the cell needs or make stuff that the body needs, because what it does is the endomembrane system will also send stuff to the rest of the body. It'll go out through the membrane into the circulatory system, especially like hormones and stuff like that. So there are a lot of organelles that are connected either through physical contact, so their membranes are actually touching each other, or there are transport vesicles that go back and forth. So you can see there that we got the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuoles, the plasma cell membrane. Okay, plasma or cell membrane, you'll see both of them. So they basically create functional compartments inside the cell. I am not going to spend a whole lot of time going over every single organelle and every single function. It is your responsibility to read chapter 4 if you don't necessarily know or remember them. Okay? So the whole purpose of basically the endomembrane system is to actually make something. Okay, whether it's making a protein, whether it's making something for for cellular division, whether it's making something that's going to secrete. So we start with the nucleus. The nucleus is where the directions come through. And then we go to, sorry, I keep hitting that. We go to the endoplasmic reticulum. So that's what ER stands for. If you want to abbreviate it, that's fine. You do not need to spell it out. So we have the rough endoplasmic reticulum or the smooth endoplasmic reticulum. And the rough endoplasmic reticulum has ribosomes on it. That's why it's rough ribosomes. And the smooth endoplasmic reticulum does not. And so these two things make different stuff. So rough endoplasmic reticulum with ribosomes on it make proteins, since that's what ribosomes do. Smooth endoplasmic reticulum does not have ribosomes on it, so it makes lipids. Okay, so it'll make a lipid, and also like calcium regulation and stuff like that. So from the endoplasmic reticulum, depending on which one, we get a transport vesicle, which basically just moves stuff around. That transport vesicle goes to the Golgi apparatus. The Golgi apparatus is basically like UPS, so it has a receiving end, the cis end, so it receives that transport vesicle, goes through all of these stacks, makes changes, edits it, so it basically gets the rough draft from the endoplasmic reticulum, edits it through the whole process of the Golgi apparatus, and then on the trans end, it ships it to wherever it needs to go, and then it releases it into a transport vesicle, which is either to its final destination, either the cell membrane to be excreted or secreted technically into like the bloodstream to go somewhere else in the body or into somewhere else in the cell. So it'll become a lysosome. All right, so that's basically the process of going through it. So all of these pieces are connected. Then we have breakdown and storage. So lysosomes, which we just briefly touched on being made through that manufacture process, is a vesicle that's basically filled with digestive enzymes. And what it does is they're the cleanup crew. All right, they will digest food, they'll clean up waste products, old organelles, they're the cleanup crew, they're the janitors of the cell. Lysol, lysosome, sure, works, right? Okay. Vacuoles are a closet, basically. It's a place to store stuff. In plant cells, they have a very large one, and it stores water, photosynthetic stuff like sugars that were made, all sorts of other things. Very large in plants. And then they're smaller in animals because animals don't necessarily need to store as much stuff, but they do actually they have smaller vacuoles. Basically, it's just a storage spot. And then contractile vacuoles is a water storage. So basically, a lot of time in freshwater protists, so there's generally single-celled organisms that live in fresh water, and they have these contractile vacuoles that absorb all the fresh water and then expel it out. It's kind of like a little water cannon. Expels it out back into the environment because otherwise the cell would burst because there'd be too much water in there. It's basically like it gives it a way to get rid of all the excess water as opposed to popping like a water balloon, like if you overfill a water balloon and it pops. So that contractile vacuole is to keep the cell from popping. Okay, last but not least, the energy conversion. We already talked about the mitochondria and the chloroplast when we talked about photosynthesis and respiration. So remember, the mitochondria is in plants and animals and goes through cellular respiration in the presence of oxygen. Okay, remember we've got the two steps that happen there. So glycolysis happens in the cytoplasm, and then we go into the citric acid cycle and the oxidative phosphorylation in the mitochondria. So that's what happens there. And then the chloroplast in photosynthetic organisms, mainly green plants, all right, photoautotrophs. Okay, so they have the chloroplast. So we already talked about those, so I'm not going to belabor that point too much. So the only thing that I wanted to add about the mitochondria and the chloroplast that we didn't really talk about is there's evidence that shows that the mitochondria and chloroplast evolved through this process called endosymbiosis. So endosymbiosis is this idea that that mitochondria and chloroplast used to be their own prokaryotes, and they were absorbed by a precursor to the eukaryote. Okay, they were absorbed by these organs or these cells. Oh, excuse me. They were absorbed by these cells long before, and instead of actually being digested, they actually started working together. Okay, and there's a lot of different pieces of evidence that show that this is probably the case. The big one is that the mitochondria and chloroplast have their own DNA, so they have their own control system. A lot of what the mitochondria and chloroplast do is regulated by themselves. So they replicate themselves. They don't need the bigger cell to tell them what to do in a lot of senses. And then they have a double membrane and some other stuff. But the big thing is that they have their own DNA. Okay, so we're going to talk a little bit more about that. All right, wrap up. Okay, I want you to create some sort of visual representation. This can be whatever works for you. You can draw a picture, you can create a table, a Venn diagram, whatever. Let your creative flag fly, okay? I want you to take this and put this together in some sort of visual representation, okay? And then upload that to Blackboard. You can either do it through like Paint, you can draw on your computer, you can take a picture of a drawing that you do on a piece of paper, okay? I will see you in class. Have a great day. Bye.